You know how large language models, LLMs, they can speak so fluently, right? But sometimes their responses are just, well, bizarre.
Yeah, totally. They might give you information that sounds really convincing,
Right.
but it's just plain wrong.
Or they just completely make things up, what we call hallucinations.
Exactly. It's this strange mix of like fluent speech, but nonsensical content.
What if that strange, sometimes unsettling fluency actually mirrors a specific human condition?
That's the really fascinating hook here.
Today, we're diving deep into a groundbreaking new study. It's from Advanced Science.
And it takes this remarkable leap comparing the internal workings, the information processing of LLMs.
With human brains, specifically brains experiencing aphasia.
Right.
So we're going to unpack the surprising similarities, the really cutting edge methods they use to, peer inside both AI and human brains.
And importantly, what this could all mean for how we understand intelligence itself,
Hmm.
both artificial and, well, biological.
Okay, so let's start with the behavior, the stuff you can actually see. That's what kicked this whole thing off.
Yeah, the observation, you ask an LLM something.
And it fires back this persuasive, fluent answer almost every time.
Look, like we said, those answers can be deeply inaccurate, riddled with those hallucinations.
It's those fluent mistakes that are so weirdly familiar.
hmm And what's fascinating is how closely that pattern, that behavior, resembles symptoms of what's called receptive aphasia in humans.
Receptive aphasia, like Wernicke's aphasia.
Exactly. Wernicke's is a key example. People with this condition, they can speak with perfectly normal rhythm. Grammar seems fine.
But the content,
The actual content of their speech often just doesn't make sense. It's like a word salad sometimes.
and crucially,
Crucially, they also have real difficulty understanding language themselves. Comprehension is impaired.
wow. So the words are there, the grammar's there, but the meaning is just lost for both the speaker and the listener.
That's a good way to put it.
That sounds eerily familiar when you think about some LLM outputs.
It absolutely does. And when you start really digging into the specifics, the parallels get even stronger.
Like what?
Well, okay, consider the repetition of non-informative words, just filler words repeated over and over.
Uh,
You see that in Wernicke's aphasia, and you definitely see it in LLM hallucinations.
okay, what else?
Then there's the LLM's sort of insensitivity to the underlying message, the context, that's very much like the language comprehension problems in receptive aphasia.
Right.
Plus, studies show LLM outputs are often structurally different from human writing. They can lack linguistic diversity, kind of repetitive phrasing.
And those sudden topic shifts. You're reading an LLM answer and suddenly it just veers off somewhere completely random.
That strongly resembles the decrease in linguistic cohesion, the way thoughts don't quite connect that you see in the speech of people with receptive aphasia.
So right off the bat, the way these AIs fail, it sounds disturbingly human in a very specific way.
It really does. And that alone is a huge insight maybe into how they're working or, well, failing internally.
So these really striking surface similarities, they naturally led the researchers to ask, a really bold question. Is there something similar actually happening inside the LLMs in their information processing? Something that mirrors human aphasia? But
Yeah.
how on earth do you even begin to compare that? A human brain and, lines of code and network weights? That seems like a massive leap.
It does. And it required a really innovative approach, methodologically speaking.
Okay.
The researchers focus on something called collective network dynamics.
Collective network dynamics.
And to actually observe these dynamics, they used a technique called energy landscape analysis.
Energy landscape analysis. Okay, that sounds like physics, not brain scans or AI.
Huh. Yeah, it does sound a bit like that.
Can you give us the lowdown? What does that actually mean for this study?
Okay, think of it like this. Imagine a ball rolling across a bumpy landscape.
Yeah.
You got hills and valleys. The valleys are what we call attractors. They're stable states where the network, or the ball in our analogy, tends to settle down.
Okay, like preferred patterns of activity.
Exactly. And the energy landscape analysis tracks the ball's movement. How long does it stay in a valley? That's an attractor. How often does it jump between different valleys?
I see. So it maps the flow and the sticking points.
Precisely. It represents the dynamic changes happening in the network over time. And the great thing is, this method is super versatile. You can use it to extract these dynamics from all sorts of time series data,
Like brain signals.
like brain signals from fMRI, yes, or even the internal activity patterns within an AI model.
Okay, so it's a bridge, conceptually. How did they apply it to human brains then?
So for the humans, they looked at resting state fMRI data. That's functional MRI scans taken when the brain is just, idling, not doing a specific task.
Resting state. Why resting?
Because those signals are thought to represent the sort of functional backbone of our cognitive abilities, the underlying wiring and dynamics.
Got it.
They gathered data from several groups. Stroke patients with 4 different types of aphasia, that's anomic, Broca's, conduction, and Wernicke's.
Okay.
Also, stroke patients who didn't have aphasia, and healthy control subjects with no stroke or aphasia.
A really comprehensive comparison.
Yeah, and this energy landscape method, it's got a solid track record. It's been used successfully to find atypical brain dynamics in conditions like autism, ADHD, depression, Alzheimer's.
So it's a proven tool for mapping human brain activity differences.
Right.
But okay, back to the AI. How do you get internal network activity from an LLM? You can't just stick it in an fMRI machine, right?
No, not quite. That's a great question.
So what do they do?
For the LLMs, they essentially probed the internal activity, the activation patterns within the network layers after the model had generated an answer.
Ah, okay. Which LLMs did they look at?
They used 4 specific ones: Albert, which is from Google, GPT-2 from OpenAI, Llama 3.1 from Meta, and LMJP3, which is a Japanese version of Llama.
Why those particular models?
Good question. They were chosen partly because they share some fundamental computational principles with how human brains are thought to process language.
Like the attention mechanism.
Exactly. They're built on the attention mechanism. They're transformer-based models. These are designs that let the models weigh the importance of different words to understand context, which is some conceptual parallels to brain processing.
Okay.
Plus, some earlier studies, particularly with GPT-2, had already hinted at some similarities between LLM activity and brain function. So there was precedent.
So models chosen for their potential architectural similarities. Once they had this internal LLM activity data, what specific measurements did they use? How did they quantify these energy landscapes?
They focused on 2 main indices, 2 key numbers.
Okay.
First was dwelling time. Basically, how long did the network's activity pattern stay settled in each attractor, each stable state or valley?
How long it lingered.
Right. And second was transition frequency.
Yeah.
How often did the activity pattern jump between these different attractor states?
How jumpy it was.
Exactly. And then to understand how polarized or varied these patterns were.
Polarized, meaning?
Meaning, did the activity cluster heavily into just a few states, or was it more evenly spread out?
They use something called Giannini coefficients.
Giannini coefficients,
Yeah.
like they use in economics for inequality.
The very same concept, yes. Applied here to network dynamics. A Giannini coefficient close to 1 means a highly polarized distribution. The network strongly prefers just a few states, like very deep valleys.
Okay.
While a value closer to 0 means a much more uniform distribution, the network explores many states more evenly.
Got it. Dwelling time, transition frequency, and Giannini coefficients to measure the shape of those distributions.
That's the toolkit.
So before they even touched the LLMs with this toolkit, they first used it to map out the human brains, right?
Yeah.
To characterize the different aphasias internally. What do they find there? Expressive versus receptive?
Yeah, they established that baseline first, and the findings were really distinct and quite revealing.
For expressive aphasia, like Broca's aphasia, where people have trouble producing fluent speech,
Right.
their brains showed shallower and more uniform attractors on that energy landscape.
Shallower and more uniform.
Meaning their internal brain activity didn't settle into really deep, distinct patterns. It was more sort of spread out, less differentiated.
Okay.
This led to relatively uniform distributions for both how long they stayed in states, dwelling time, and how often they jumped, transition frequency.
And the Gini coefficient.
Their dwelling time Gini coefficients were significantly lower than the healthy controls, less polarized. And interestingly, this decrease actually correlated with how severe their speech fluency problems were.
Wow. So the flatter landscape matched the difficulty speaking.
Seemed to, yes.
Mm.
But for transition frequency, how often they jump between states, their Gini coefficient wasn't significantly different from controls.
Okay, so that's expressive aphasia. What about receptive aphasia, the one that looked behaviorally similar to the LLMs?
Here's where it gets really interesting. In stark contrast, brains with receptive aphasia, like Wernicke's and conduction aphasia. They showed a much more diverse energy landscape. Attractors with really varied depths, some very deep, some quite shallow.
So a more rugged landscape.
You could say that. And this generated highly polarized sort of bimodal distributions for both dwelling time and transition frequency, much less uniform.
Okay. How did their Gini coefficients compare?
Well, for dwelling time, their gene coefficients were only marginally different from the healthy controls. Not a big difference there. But their transition frequency Gini coefficients were significantly larger than the controls, much more polarized, jumping between states.
Larger Gini for transitions. What does that mean functionally?
It suggests their internal brain states were maybe getting stuck in very specific isolated patterns or jumping erratically between a few dominant states. And critically, this atypically high transition Gini coefficient was directly correlated with how poor their language comprehension was.
Whoa. So low Gini for dwelling time and expressive aphasia relates to speech production problems. High Gini for transition frequency and receptive aphasia relates to comprehension problems.
Exactly. These two Giannini coefficients, one for dwelling time, one for transition frequency, they almost became like an internal signature.
A diagnostic fingerprint based on network dynamics.
Pretty much. They proved to be really effective indices for classifying the different types of aphasia just based on these internal dynamics. You could plot them on a graph and see distinct clusters for expressive, receptive, and healthy controls.
That's incredible. A powerful tool just from the brain dynamics.
Yeah.
So now we get to the LLMs. With this clear classification map for human brains, the researchers finally turned this analysis onto Albert, GPT-2, Llama, and the others. What did their internal dynamics reveal?
Right. This is the moment of truth, so to speak.
What did they find?
Okay, first off, a general observation. The energy landscapes in all the LLMs they looked at had, on average, deeper attractors than the human brains.
Deeper valleys, meaning they settle more rigidly.
That's the interpretation, yes. A tendency for their internal states to lock into very defined, perhaps more rigid patterns compared to the human brain studied.
Interesting. And what about the Gini coefficients? Did the LLMs cluster with any of the human groups?
This is the crucial finding. When they look at the Gini coefficients for the LLMs, both the dwelling time Gini and the transition frequency Gini were significantly larger than those seen in the healthy human controls and in the expressive aphasia group.
Okay, so different from healthy brains and from Broca's type aphasia. What about receptive?
Here it is. There were no distinct significant differences seen between the LLMs and the receptive aphasia group.
No difference. Seriously?
Seriously. For example, comparing GPT-2 directly with the Wernicke's aphasia group, their dwelling time Gini coefficients showed only marginal, non-significant differences. And the same held for transition frequency.
So, the internal processing patterns of these big AI models like GPT-2 and Albert, they looked statistically much, much more like human brains with receptive aphasia than they did healthy brains or brains with expressive aphasia.
That's exactly what the data shows.
That's a profound parallel, mind-blowing, actually.
It really is. It indicates this very significant similarity, not just in outward behavior, but in how they seem to process information at the internal network dynamics level. It mirrors the collective brain dynamics seen in receptive aphasia.
Okay, this is incredibly compelling. But it also brings up a really important question, one I'm sure you're thinking about too. Does this mean LLMs are sick? Or, that they have a brain injury like humans with aphasia? We're not saying that, are we?
No, absolutely not. And that's a critical point this study is very careful to make. It's about clarification.
Right.
While the similarities in behavior in these internal processing dynamics are striking, really striking, the study acknowledges a crucial limitation. The reasons behind the inaccurate responses and hallucinations in LLMs might be very different from the underlying causes of receptive aphasia symptoms in humans.
Okay, so different roads leading to a similar-looking destination, maybe.
That's a good analogy, yes.
Can you elaborate on what those different roads might be? What causes LLM weirdness versus human aphasia?
Certainly. So for LLMs, hallucinations and inaccuracies are generally thought to be linked to the data they were trained on. Inaccurate or incomplete knowledge picked up during pre-training.
Bad data in, weird output out.
Essentially. And we know that improving the quality of that pre-training data tends to reduce hallucinations. This points towards what the study calls a problem of shallow language matching.
Shallow matching.
Like, they get good at predicting the next word based on patterns, but maybe without a deep, robust understanding or grounding of the meaning.
Okay. And receptive aphasia.
Receptive aphasia in humans, conversely, is directly linked to physical damage. Stroke-induced damage to specific brain-wide networks that are absolutely essential for properly connecting words and sentences to their actual meaning.
So one is potentially a kind of knowledge or architectural limitation in the AI. The other is actual structural brain damage.
Very different causes.
Fundamentally different causes, yes, that's the hypothesis.
That's a really important distinction. Are there other differences maybe in the language itself, even with these similarities?
Yes, definitely. That's another nuance. While Wernicke's aphasia involves those comprehension problems and word repetition, the actual speech produced by someone with Wernicke's often shows less linguistic cohesion, less complexity than even a hallucinating LLM.
Less coherent than an LLM.
Often, yes. In fact, some of the really advanced LLMs, like GPT-4, can actually improve the linguistic cohesion and flow of essays written by humans.
Right, so LLMs can be too coherent in a way, even when they're wrong, compared to the more fragmented speech in aphasia. That's another subtle difference, yes. The texture of the output can differ, even if the underlying dynamic pattern shows similarities.
Okay, so given all these complexities, these similarities and differences, where does research need to go next to really unpack this?
Well, the study itself emphasizes the need for much more research to clarify both sides of the coin, the similarities and the differences,
How?
by really scrutinizing the finer details, looking deeper into the information processing pathways in both systems, and carefully analyzing the precise nature of the linguistic behaviors that result. The study's really just the beginning.
A fascinating beginning, though. So after this really deep dive, what's the biggest takeaway for us, for the listeners? What's the most exciting or maybe provocative idea to carry forward?
I think the most profound concept, the really novel idea proposed here is this. We might potentially be able to assign a kind of diagnosis to an LLM.
A diagnosis for an AI.
Yes. By comparing its internal network dynamics, the Gini coefficients, the energy landscape, with the patterns we see in typical or atypical human brains.
Wow. So instead of just judging an LLM by what it says, its output, we could look at how it's processing information internally.
Exactly.
To infer something about its performance, its capabilities, maybe even its limitations or biases.
Precisely. This whole approach could lead to an entirely new way to characterize these powerful AI models. It might allow us to infer their performance, their tendencies, maybe even their reliability before we even use them extensively or sift through their outputs.
Imagine diagnosing an AI's potential flaws just by looking under the hood at its dynamics. That's powerful.
It's a really intriguing possibility.
What a mind-bending journey that was into these hidden parallels between AI and, well, the human brain itself.
It really makes you think.
We've seen how digging into these internal network dynamics can throw up these completely unexpected similarities between LLMs and human conditions like receptive aphasia. It definitely forces us to rethink some assumptions about intelligence.
It's a profound reminder, I think. Even as AI gets incredibly advanced, maybe there are some fundamental principles of how information is processed, how systems learn and fail, that actually bridge the gap between biology and silicon.
Offering new ways to understand both?
Potentially, yes. New ways to understand the brain and new ways to understand AI.
So here's the final thought to leave you with. What stands out most to you from this deep dive? Is it the possibility that we might one day have, like, AI neurologists diagnosing an LLM's cognitive state from its internal dynamics?
Or does this research mainly just highlight the incredibly surprising, sometimes baffling, and often unpredictable ways that intelligence, or something like it, can manifest, whether it's grown or built?
It really makes you pause, doesn't it? And consider how much we still don't understand about the very nature of intelligence itself.